Good morning, Doc, and to my classmates. So, I am tasked to report about proteins and its level of structure. So, ano nga ba ang proteins? What are proteins? So, it says here proteins are large molecules. They are also made up of chains of amino acids. They are found in every cell in the body, and they are involved in most of the body's functions and life processes. And the sequence of amino acids is determined by DNA. So proteins are large and they are essential molecules found in every living cell in the body. So gawa sila sa isang napakahabang chains of smaller units called amino acids, which are like the building blocks, or it's not like, probably the building blocks of proteins. So alam naman natin na kapag building blocks, they are like the main components of proteins. Now, ang mga amino acids na to are linked together in a specific order, and the way they are arranged determines the protein's shape and function. Now, let us always remember that every part of our body, mula sa ating muscles, sa skin, to our hair and organs, they contains proteins. And these proteins are crucial for many functions that keep us alive and healthy. For example, we have enzymes. So we all know that enzymes help our body speed up chemical reactions like breaking down food during digestion. And another examples are structural proteins and transport proteins. So what's the difference between the two? Kapag sinabi nating structural proteins, this gives our body support and strength, like the keratin in our hair and collagen in our skin. While for transport proteins, they carry important molecules around our body, such as hemoglobin in your blood, which carries oxygen. So, in short, napakahalaga ng function ng proteins for almost every process in our body, from growth and repair to carrying oxygen and fighting off infections. And the unique sequence of amino acids in each protein is determined by DNA that allows it to work properly and keep us healthy. Now, ang nakikita natin ngayon is the structure of amino acids. So, ang lahat ng amino acids ay may parehong simpleng structure. At the center of each amino acid, you can see here, this is called the alpha carbon. Attached to the alpha carbon are four groups. So we have hydrogen atom. We also have a carboxyl, an alpha carboxyl group, an alpha amine group, and an R group, also called the side chain. So, ang alpha carbon, carboxyl at amine groups ay pareho sa lahat ng amino acids, pero ang R group ang naiiba sa bawat amino acid. Dahil sa iba't ibang R groups, nagiging posible para sa amino acids na magkaroon ng maraming iba't ibang mga tungkulin, mula sa pagbuo ng mga enzymes hanggang sa pagsuporta sa structure ng mga cell. So sabi natin kanina, amino acids are the building blocks of proteins. And nutritionists categorize them into two main groups based on how the body obtains them. So these are essential amino acids and non-essential amino acids. So this classification isn't actually related to their structure, but more of how the body manages to get them or make them. So let's start first with the essential amino acids. So what is all about essential amino acids? So these are the amino acids that the body cannot synthesize on its own. So it cannot be synthesized by the body. This means that we must get them through our diet. So from the foods we eat, like meat, fishes, eggs, beans, and nuts. So there are 9 essential amino acids for humans. So we have here histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So the number and types of essential amino acids can vary between species. For example, what is essential for humans may not be the same for plants or animals. In humans, the needs for essential amino acids can also differ depending on age. Example for children who are still growing, they often need more essential amino acids than us adults. For example, we have the arginine. So this arginine is essential for children, but not actually necessarily for adults because our body can produce enough on their own. Now, ano nga ba ang function ng arginine na to? So, ang arginine, this provides a wide range of health benefits, including better blood circulation, it enhances immune function, faster wound healing, detoxification of ammonia, hormone regulation, and support for muscle growth and fertility. So that's for essential amino acids. Next, we have non-essential amino acid. So what is a non-essential amino acids? So non-essential amino acids, these are those that our body can synthesize. So it can be synthesized from other compounds. So we don't need to rely on diet alone to get them. Even though they are called non-essential, they are still very important for normal body functions. Examples include alanine. So this alanine, it provides energy for muscles. It also supports glucose production, it detoxifies ammonia, and aids immune function. Also, we have asparagine. So, asparagine is vital for protein synthesis, nervous system function, cell growth, and nitrogen transport. And we also have glutamine. So, glutamine supports immune cells, it maintains gut health, it helps in nitrogen transport, it aids muscle recovery, and regulates acid base balance. So our cells can make these amino acids by converting other amino acids or through metabolic processes. So if your diet is lacking in this, your body can actually compensate. So that's how essential amino acids and non-essential amino acid functions. Okay, now amino acid is also categorized based on our group properties. So we have here the nonpolar group, the carboxyl group, the amine group, aromatic group, hydroxyl group, and others. Okay, so let's start with nonpolar amino acids. So ano nga bang nonpolar amino acids? So ito yung mga amino acids na ang R group or yung side chain na sinasabi natin kanina ay hindi naglalaman ng mga kargang electrical or polar bonds. So with this, ang mga nonpolar amino acids ay hydrophobic. Ibig sabihin, hindi sila madaling humalo o makipag-ugnayan sa tubig. So in fact, sila ay madalas na matatagpuan sa loob ng mga protein structures. So paano sila gumagana? Sa mga proteins, ang mga nonpolar amino acids ay tumutulong sa pagbibigay ng structure at stability sa protein. So sa loob ng isang protein, ang mga hydrophobic na bahagi ay madalas na nagtatago sa loob ng folded protein. Habang ang hydrophilic or yung mga water-loving part ay nakaharap sa labas na kung saan ito ay nakikipag-ugnayan naman sa tubig or iba pang polar molecules sa environment. So what are the functions for each non-polar amino acids? So let's start first with alanine. So alanine is one of the most abundant amino acids found in proteins, ranking second only to leucine in occurrence. So a D form of the amino acid is also found in bacterial cell walls. So if we can see here, ito ang structure ng alanine. So um, alanine could be found in bacterial cell wall. Next, we have glycine. So glycine is the amino acid with the shortest side chain, having an R group consistent only of a single hydrogen. So its Small side chain allows it to readily fit into both hydrophobic and hydrophilic environment. So proline naman is the only amino acid found in proteins with an R group that joins with its own alpha amino group, making a secondary amine and a ring. So proline is a non-essential amino acid. So it says here it's a disruptor of alpha helices and beta strand. So it is a disruptor of alpha helices and beta strands. Ibig sabihin, ito ay isang amino acid o molecule na sumisira o nagpapabago sa normal na forma ng alpha helix at beta strand na siyang nagdudulot ng baluktot o kawalan ng regular na pagkakaayos ng protein structure. Next, we have isoleucine. So isoleucine is an essential amino acid. It has a hydrophobic side chain and is also chiral in its side chain. So kapag sinabi nating chiral, so kapag sabi nating chiral, when a molecule does not coincide with its mirror image. So that's for isoleucine. Next, we have leucine. So leucine is a branched chain amino acid that is hydrophobic and essential. So leucine is the only dietary amino acid reported to directly stimulate protein synthesis in muscle. So when we talk about leucine, we have to always remember that it stimulates protein synthesis in muscle. Now next we have valine. So valine is an essential amino acid, non-polar amino acid synthesized in plants. So it is noteworthy in hemoglobin when it replaces glutamic acid at position number 6. It causes hemoglobin to aggregate abnormally under low oxygen conditions, resulting in sickle cell disease. Lastly, we have methionine. So it says here it is an initiator of amino acid in protein. So methionine. So kapag sinabi ng methionine, it is an essential amino acid that is one of two sulfur-containing amino acid. So it is the initiator amino acid in protein synthesis because it is the first one incorporated into protein change. Ibig sabihin ng initiator ay ang unang amino acid na naidadagdag sa mga protein chains sa simula ng protein synthesis. Okay, so let's have carboxyl amino acids. So let's start with aspartic acid. Now, ang aspartic acid is an unessential amino acid with a carboxyl group in its R group. So aspartic acid's side chain is negatively charged at physiological pH. Ang aspartic acid ay may mahalagang papel sa protein synthesis. It acts as a neurotransmitter for energy production, nitrogen transport, regulation ng acid-base balance, at synthesis ng iba pang amino acids. So ito ay isa sa mga non-essential amino acids na tumutulong sa maraming metabolic processes sa katawan natin. Next, we have glutamic acid. When we say glutamic acid, it is a neurotransmitter and has an R group with a carboxyl group that readily ionizes. So ang glutamic acid ay isang mahalagang amino acid na tumutulong sa pagpapadala ng mga signals sa utak natin, sa pagbuo ng proteins, sa paglikha ng energy, at pagsasaayos ng nitrogen at acid-base balance sa katawan natin. Now let's have amino acids. So we have here arginine, histidine, and lysine. So arginine is an amino acid that is, in some cases, essential, but in some cases, non-essential. Example, premature infants cannot synthesize arginine. In addition, surgical trauma, sepsis, and burns increase demand for arginine. So most people, however, do not need arginine supplements. Arginine side chain contains a complex guanidinium group with a pKa of over 12, making it positively charged at cellular pH. Next, we have histidine. So, histidine is the only one of the proteinaceous amino acids to contain an imidazole functional group. It is an essential amino acid in humans and other mammals. So, histidine is a precursor to histamine, a vital compound involved in immune responses, gastric acid secretion, and neurotransmission. Histamine also plays a crucial role in allergic reactions and inflammation. Next, we have lysine. So, lysine, it has an R group that can readily ionize with a charge of positive 1 at physiological pH. So, it is used to flag proteins for export from the cell. So lysine is often added to animal feed because it is a limiting amino acid and is necessary for optimizing growth of pigs and chicken. Next, we have aromatic amino acids. So we have here phenylalanine, tryptophan, and tyrosine. So, what is phenylalanine? So, phenylalanine is a component of the aspartame artificial sweetener. So, aspartame is a low calorie artificial sweetener that is widely used in food and beverage products such as diet sodas, sugar free gum, and various low calorie snacks. So, it is approximately 200 times sweeter than sucrose or our table sugar, which allows manufacturers to use smaller amounts to achieve the desired sweetness. Now, we also have tryptophan. So, tryptophan, though reputed to serve as a sleep aid, there are no clear research results indicating this. So, tryptophan is a crucial for the synthesis of serotonin in the body. Once ingested, tryptophan is converted into serotonin, which is then transformed into melatonin, the hormone responsible for sleep wake cycle. So, this biochemical pathway highlights how tryptophan can directly influence sleep patterns. Research on tryptophan's effectiveness as a sleep aid has produced mixed results. So, some studies suggest that supplementation with tryptophan can improve sleep quality and reduce the time it takes to fall asleep. So, while others show minimal effects, however, many experts agree that tryptophan may be more effective when combined with carbohydrates, as carbohydrates can facilitate the transport of tryptophan across the blood-brain barrier. Next, we have tyrosine. So, tyrosine, it is a target for phosphorylation in proteins by tyrosine protein kinases and plays a role in signaling processes. Tyrosine is also a precursor of thyroid hormones and melanin. So, given its role in the synthesis of thyroid hormones and melanin, tyrosine is crucial for maintaining not only metabolic health but also skin and hair pigmentation. Ensuring adequate intake of tyrosine, rich foods such as dairy products, meat, fish, eggs, nuts, and soy products can support these essential functions. Last group, we have hydroxyl amino acids. So hydroxyl acids, we have serine, threonine, and tyrosine. So serine is a precursor of many important cellular compounds, including purines, pyrimidines, sphingolipids, folate, and of the amino acids glycine, cysteine, and tryptophan. So, serine is a versatile amino acid that plays a vital role in protein synthesis, metabolism, neurotransmitter production, immune function, and cell signaling. So while the body can produce serine, it is also found in various dietary sources, including meat, fish, dairy products, eggs, nuts, and soy products. So next we have threonine. So threonine, it is one of the three amino acids bearing a hydroxyl group. Serine and tyrosine are the others, and as such is a target for phosphorylation in proteins. In some applications, it is used as a prodrug to increase brain glycine levels. Ibig sabihin, na ang ilang mga compound ay ginagamit bilang prodrugs upang madagdagan ang antas ng glycine sa utak. So ang pagtaas ng glycine levels ay maaaring magdala ng mga benepisyo sa mental health at cognitive function. Next, we have tyrosine. Tyrosine is a versatile amino acid also that plays critical roles in neurotransmitter production, thyroid hormone synthesis, melanin production, and protein synthesis. Its potential effects on cognitive function and mental health, as well as its antioxidant properties, make it an important nutrient for overall health. Now, how does peptide bond form? Well, an amino acids are organic compounds that serve as the building blocks of proteins. So while they have various functions in the cell, like signaling metabolism and serving as precursors for other molecules, their primary role is actually to be linked together to form proteins. So peptide bonds are the specific covalent bonds that link amino acids together. So the formation of a peptide bond occurs through a dehydration synthesis reaction, also called condensation reaction, in which the carboxyl group or the COOH one amino acid reacts with the amino group or the NH2 of another amino acid. So during this reaction, a molecule of water is released. So this process can be summarized as amino acid 1 or the COOH plus amino acid 2 or the NH2 that leads to dipeptide or CONH plus water. So in this animation, we can see how a peptide bond is formed. So we have here the primary structure of a protein. So from gene to protein. Now the relationship between DNA, genes, and proteins is crucial for understanding how traits are expressed in living organisms and how cellular functions are regulated. So the role of DNA in determining the primary structure is that the DNA or the deoxyribonucleic acid strands are made up of units called nucleotides. So each consisting of a sugar, a phosphate group, and a nitrogenous base. So there are four types of nitrogenous base in DNA. We have adenine, which um, with A as the symbol, thymine with D as the symbol, cytosine with C as the symbol, and guanine as G as the symbol. So the specific sequence of these bases encodes the genetic information. So within the DNA molecule, we have segment known as genes that serve as functional units. So a gene is a sequence of nucleotides that contains the instructions for synthesizing a specific molecule, usually messenger RNA. So the process by which the information in a gene is used to synthesize protein is known as gene expression, and it occurs through two main steps. We have transcription and translation. For transcription, during this process, the DNA sequence of a gene is transcribed into mRNA. So the mRNA strand is complementary to the DNA template strand and carries the code for the nucleus to the ribosomes, where proteins are synthesized. For translation, once the mRNA reaches the ribosome, it serves as a template for the synthesis of a polypeptide chain. So the ribosome reads the sequence of mRNA in sets of three nucleotides called codons, each of which corresponds to a specific amino acid. For transfer RNA, molecules transport the appropriate amino acids to the ribosome, where they are linked together by peptide bonds, forming a polypeptide. So the actual order of amino acids in the primary structure of a protein is dictated by the sequence of nitrogenous bases in the corresponding gene. So each amino acid has unique properties that influence how it interacts with other amino acids in the chain. So these interactions can lead to the formation of various structural elements within the protein, such as alpha helices and beta pleated sheets, as well as more complex three-dimensional shapes. So the final shape of a protein is crucial for its function, as the specific arrangement of amino acids determines the protein's ability to interact with other molecules in the cell. For example, enzymes rely on their specific shape to bind to substrates, while antibodies must have a precise structure to recognize and neutralize pathogens. Next, we have the secondary structure of protein. So if you can see here, that involves alpha helix and beta pleated sheet. So here's a step-by-step process on how alpha helices are formed in the secondary structure of protein. So first, we have polypeptide chain folding. So a polypeptide chain folding is a linear sequence of amino acids linked by peptide bonds. In the secondary structure, the chain starts to fold in specific ways to form regular repeating patterns. So paulit-ulit lang siya. Number two, we have hydrogen bonding. So the alpha helix is stabilized by hydrogen. Hydrogen bonds between the backbone atoms of the polypeptide chain. So these hydrogen bonds form between the oxygen atom of the carbonyl group, the oxygen atom of the carbonyl group, and the hydrogen atom attached to the nitrogen group. So these bonds occur between amino acids that are four residues apart in the sequence. So these bonding patterns pulls the change into a helical shape. Number three, we have right-handed coil. So the alpha helix is typically a right-handed spiral or coil. This means it twists in a clockwise direction as you move along the polypeptide chain. So it's a clockwise direction. Kung makikita natin pag ganyan siya. And how many residues per turn? So in a typical alpha helix, there are 3.6 amino acids per turn of the helix. So this regular structure is a result of the consistent hydrogen bonding and the arrangement of amino acids in the chain. Alright, and then number five, we have side chains point outward. So the R group side chains of the amino acids in the alpha helix extend outward from the helix. These side chains do not participate in the hydrogen bonding that stabilizes the helix but can influence the overall stability and function. So what about first secondary structures of protein? So we have here six processes on how a pleated sheet is formed. So first we have polypeptide chain. Formation of polypeptide chain. So the beta pleated sheet is formed from one or more polypeptide chains. A polypeptide chain consists of amino acids linked by peptide bonds in a linear sequence. So unlike alpha helices, the chain is typically more extended and coiled. The beta pleated sheet consists of multiple beta strands. So these strands are segments of the polypeptide chain that run alongside each other, either in the same direction, parallel, or in opposite directions, anti-parallel. So when we say parallel, the beta strands run in the same N terminus to C terminus. While for anti-parallel, the beta strands run in opposite directions relative to one another. And then there would be hydrogen bonding. So the beta strands are held together by hydrogen bonds. So sa hydrogen bonding, the beta strands are held together by hydrogen bonds between the backbone atoms of the polypeptide chains. So the hydrogen bonds form between the carboxyl oxygen of one amino acid and the amide hydrogen of another amino acid on an adjacent strand. So there we have here. So these hydrogen bonds form across the strands, creating a stable sheet-like structure. So in antiparallel sheets, the hydrogen bonds are more linear and therefore stronger. So while in parallel sheets, they are slightly angled, which makes them a bit less stable. So next, we have pleated appearance. So the pleated appearance of the sheet arises because the beta strands are not completely flat. So instead, the backbone of the strand alternates between pointing slightly up and down, creating a rippled or pleated shape. So the side chains or the R groups of the amino acids also alternate, pointing above and below the plane of the sheet. So this alternating pattern helps maintain the stability of the sheet and allows for specific interaction with each other molecules. Next, we have the parallel versus antiparallel arrangement. So the antiparallel beta sheets, the beta strands run in opposite directions. So this arrangement is more stable because the hydrogen bonds between the hydrogens are more direct and linear. For parallel beta sheets, the beta strands run in the same direction. So the hydrogen bonds are slightly angled, making this arrangement less stable than anti-parallel sheets. And lastly, the stabilization of the beta sheet. So the beta pleated sheet is further stabilized by the regular hydrogen bonding. So we have here across the strands. So the sheet-like structure is also stabilized by interactions between the R groups of the amino acids in different strands. For example, the hydrophobic side chains may interact with each other, adding stability to the sheet. So next we have tertiary structure. So how does tertiary structure is being formed? So we have the first step, the secondary structure folding. So the protein begins as a linear polypeptide chain made of amino acids, folds into its secondary structures, or the alpha helices and beta sheets, through hydrogen bonding in the backbone. So these secondary structures provide the initial organization of the protein, but they are not yet in their final functional form. So next we have interactions between R group side chains. So the tertiary structure is mainly formed by interactions between the side chains or the R groups of the amino acids in the polypeptide chain. These interactions fold the protein into a complex 3D shape. Several types of interactions are involved in the processes. So we have the hydrophobic interactions. So for hydrophobic, sabi natin, when you say hydrophobic, it's a water-fearing side chains of amino acids. So they tend to cluster together inside the protein away from the aqueous or the water-based environment. So this drives the folding of the protein by pushing nonpolar side chains towards the core of the proteins. And then next we have hydrogen bonds. So the hydrogen bonds form between polar R groups or between polar side chains and the backbone of the protein. So these bonds help stabilize the folded structure. Next we have ionic bonds. So ionic interactions occur between positively and negatively charged side chains. Example, between lysine and glutamic acid. So these strong electrostatic attractions further stabilize the protein structure. And disulfide bridges. So a special type of bond called a disulfide bond or bridge forms between the sulfur atoms in the side chains of cysteine residues. This covalent bond is particularly strong and helps stabilize the tertiary structure by linking different parts of the polypeptide chain. And then lastly, we have the van der Waals interaction. So in van der Waals forces, the forces are weak interactions between nonpolar side chains. So even though these forces are weak, when combined, they contribute to the overall stability of the protein. Number three, process on how a tertiary protein structure is being formed would be folding into a compact shape. So as the chains interact, the protein folds into a compact globular shape. Hydrophobic residues are usually buried inside, while hydrophilic residues are exposed on the surface, interacting with the aqueous environment. Lastly, we have domain formation. So large proteins often fold into several domains, which are distinct regions of the protein that can function independently or contribute to the overall function of the protein. So each domain is typically a combination of alpha helices and beta sheets folded together. So if we can see in the picture, this represents how a protein is being structured from primary up until the quaternary structure. So what are the words we have to always remember when it comes to quaternary structure of protein? So we have oligomeric, which means two or more polypeptide chains or subunits, and then homotypic, almost identical subunits, heterotypic, different subunits. So this defines the arrangement and position of each subunit in an intact protein. So what are the different polypeptide subunits? So we have here homomeric proteins. So when we say homomeric proteins, if the subunits are identical, so the protein is called homomeric. While heteromeric proteins, if the subunits are different, the protein is heteromeric. So for the subunit organization, this is the way in which the subunits are arranged in the quaternary structure. So it can vary widely depending on the protein. So some common arrangements include dimers or two subunits. Example, we have certain enzymes, trimers that has three subunits, and tetramers that has four subunits. Example, we have the hemoglobin. Oligomers, more than four subunits, sometimes hundreds or even thousands in large protein complexes. Examples, we have ribosomes. Examples of proteins with quaternary structure, we have hemoglobin. So a tetramer consisting of two alpha and two beta subunits responsible for oxygen transport in the blood. We also have collagen, a fibrous protein made up of three polypeptide chains wound together like a rope, providing structural support to tissues. Immunoglobulins, or the antibodies, composed of four subunits that has two heavy chains and two light chains, which work together to recognize and bind specific antigens in the immune responses. So the quaternary structure of proteins is the result of the assembly of multiple polypeptide chains into a functional protein complex. These interactions are stabilized by non-covalent forces like hydrogen bonds, hydrophobic interactions, ionic bonds, van der Waals forces, and sometimes covalent disulfide bonds. So the quaternary structure is crucial for the proper functioning of many proteins, enabling complex biological processes such as oxygen transport, immune responses, and enzymatic regulation. So that's it for proteins and its structure.